Teachers, would you like to come in and see? A warm welcome from NJC and Singapore to our class. Today, just to let you know what we are doing or what we've been doing so far, we started our second last topic on genetics. So that's where we are. For our friends, welcome. I'm not sure, do you take biology? And back in Vietnam? Okay, don't do biology. Alright, so I will need all your help to get them up to speed. As for the rest of you, send by your iPad as well as your SLS. We're going to continue on with the previous lesson. Okay, today we are going to go to tab A2. Tab A2, that's where we are at. We are at tab A2. Okay, to paint the scope for today, as usual, we start with a bit of recap. After that, then we're going to the crux of today's lesson. We're going to learn how to draw genetic diagrams. This is your bread and butter for this particular chapter. Knowing how to draw genetic diagrams will help you solve a lot of the puzzles. Some of the puzzles you tried last week. Okay? First and foremost, we'll do a little bit of recap. Last week we played the game to genetics. Remember? Through playing the game to genetics, we learned about some of the vocabulary defined in this chapter. So the first thing we'll do today is we will recap all the vocabulary. I'd like to show you 3 digits that you have encountered last week. Digit 1, 2, and 3. You also see their genotypes. Yeah? Some of the vocabulary we're going to recap this morning. First and foremost, genotype. I'd like you to think for yourself, in your own words, how will you describe what genotype is? Second, phenotype. Same thing, in your own words, how will you describe what phenotype is? You can refer to the diagram here to get some inspiration before you share your view. Okay, ready? What is genotype? What is phenotype? In your own words. Okay, let's begin first with genotype. What is genotype? Kira, in your own words. Okay, Kira says genotype refers to the 2 types of alleles. What do you say? In a gene. Okay, in a gene. Any other definition? In your own words. Ella? So you say again. Oh, the genes that are below the surface. Okay. Ella says, perhaps genotype refers to the genes that are present below the surface. Where? Going through your intestines, going through the blood. Okay, genotype is not something that appears on the outside. As opposed to, it sounds like you're comparing with something. Okay, as opposed to the phenotype. Okay. One more, let's take one more. What is your definition of a genotype? Alpha? Oh, alpha without notes. Alpha is almost go forward what we went through in class. Alpha states combination of alleles. Okay? Out of these three, I would say alpha kind of captures the spirit of what genotype means. If we look at any organism, at any one point in time, they have 2 copies of every version of the gene, right? And I think that's what Iran is trying to do too. Everybody has kind of 2 types, but not necessarily type. For example, if you look at this particular pigeon here, she has the same type of allele, right? So I would say what Iran has got right in this aspect is that there are 2. Everybody will have 2 copies. 2 copies of alleles present in a gene. Well, an allele is nothing more than a different version of the gene. So one would say that you have 2 alleles present, 2 copies of an allele present per individual, okay, for that particular gene. So let's remove this part. And that says genes present below the surface. I think what Ella is trying to encapsulate here is that, when you look at alleles and genes, not necessarily something you can see. But what they encode for through transcription and translation are things you could see in the surface. That's why you are trying to relate to the phenotype. So what is a genotype? It's really nothing more than a combination of alleles. For example, I could ask, what is the genotype of this particular pigeon here? And that's where we lead to the next set of vocabulary. Let's do a quick recap of these few words. There's homozygous, dominant, homozygous, recessive. What's the last kind of genotype you can encounter? Heterozygous. And encapsulated in the name, homo means same. Same of what? The dominant allele. Same of what? The recessive allele. Hetero means not the same. So you have different combinations in that pair, 2 different alleles present. Okay, so if I were to ask you, what is the genotype of this particular pigeon with the no crest phenotype? What will your response be out of A, B, C? Okay, think for yourself. What is the genotype of this pigeon which has the no crest phenotype? Ready? So wait. Heterozygous. Okay, heterozygous. Because, so wait, why? Okay, so we observe that one of each allele present is not the same. The next tricky part is this. We observe that these 2 pigeons have a homozygous genotype. Now, how do you tell which allele, the crest or no crest, is the dominant or recessive one? The truth is this table gives you enough information to know which allele is the dominant, which allele is the recessive. Can you tell? Can you tell? Okay. Teacher is nodding ahead, behind. Teacher, which allele is the dominant allele and what is your reasoning behind it? Okay. Teacher, you are saying we should focus our attention on this pigeon, is it? The second one? Okay. Second one meaning? Second column. Column is row. Okay, second row. Okay, second row. What about the second row? Okay, teacher asks, is the dominant one no crest? You all agree? Everyone agrees. What's your reasoning why you think the no crest one is the dominant allele? What's the pigeon that came out? There's no crest. Yeah? So the learning point here is that, how do you tell if an allele is dominant? You can tell if, when you look at the heterozygous combination, genotype, the allele that reflects itself on the surface, that would be the dominant one. Because the other one got marks. Yeah? So it is very intuitive for all of you to come to this conclusion. But how you write it out in words, make use of all these vocabulary. Okay? Words like homozygous dominant, homozygous recessive. I observed that this pigeon has a heterozygous genotype, and the phenotype that shows is no crest. Therefore, the no crest allele must be the dominant allele. This is how you can string all the words together in a sentence. Okay? So that's where we left off last week. We came across words like genotype, phenotype, which is the traits you see on the surface. We came across words like homozygous dominant, homozygous recessive, heterozygous. These are words you use to describe genotypes. Phenotypes, I would say it changes according to the context. One time you may get flowers, then you use words like purple flowers or white flowers. You may get pigeons, you may get dogs, you may get bees. So the context will change. But these words, this vocabulary shouldn't change regardless of organism. You can use these words, vocabulary repeatedly. Okay, so that is a quick recap of last week. Then we're going to dive into your bread and butter of this topic, drawing genetic diagrams. I'd like to provide you a quick sample. If you refer to A2 of your SLS, you will see one of the activities asks you this question. A pigeon, which is heterozygous for the crest gene, mates with another pigeon which is also heterozygous for the crest gene. Okay? And the question that asks, can you tell me what is the probability that an offspring with the no crest will get the no crest phenotype? Okay? So digest this question. Okay, right now, I'm going to run through the steps that we would take to draw a genetic diagram. Genetic diagram is the biologist's toolkit. It is a model that we use to try to solve puzzles, try to understand where you are coming from when you make your justifications. So I'd like to refer you, so you must keep this question in mind. That is the backdrop of it. But as we go down, this is what we'll be able to draw by the end of the day. Okay? I'd like to show you it's not very difficult. It is things you already implicitly know, except now we're going to put it in a model so that we can share with the teacher or anyone that is looking at your justification in a much clearer way. It is a visual representation of what is already in your mind. So let's begin. I'm going to run through all the steps. So as you go through the steps, may I get you to join along with me? What you can do right now is, eventually I'd like you to submit this. So what you could do, you download this image first, join along with me, use your pencil. I'd like you to fill in the blanks with me, and later on you'll try from scratch how to draw a genetic diagram. I've provided you a template, but in this template we are going to draw and fill in blanks together. Okay, download this image first. Oh, some of you are very fast. You see, I tell you, this is intuitive. Some of you already know where to fill in the blanks here and there. Okay, when we look at a genetic diagram, the very first thing we need to do as a biology student is to define what the alleles are. Let's look at the very first line up there. It says, let n. So step one, you need to do is to define your alleles. To be honest, perhaps this is the most challenging part. But once you get this right, everything else will kind of flow from there on. Let's read the first sentence. Let n be the dominant allele for no crest, which is what Chen has already said. No crest is the dominant allele. And let small n be the recessive allele for crest trait. You observe the way we use alphabets. When it comes to the dominant allele, we use a capital alphabet. When it comes to the recessive allele, we use the lower caps alphabet. Observe that. What alphabets do you use? Actually, it is your choice. Okay? It is your choice. But I would just like to say, there are some alphabets that are better than others. Why? For example, when you look at alphabets, big caps, small caps, you notice some of the alphabets, whether big or small caps, look exactly the same. For example, maybe you give me some example of alphabets that look the same when you look at big or small caps. S, okay, so it says S. Yeah? So let the dominant allele be big S, let the recessive allele be small s. I tell you, over time as you're rushing, you cannot even tell which is big or which is small anymore. And we observe this happening. Okay, let's list down of the other alphabets you cannot work with. O, okay, O is not a very good one. Anything else? X, X is not a good one. But we can use it for sex chromosomes, right? Because it's either X or Y chromosome, but not good for alleles, because big X, small x, not much of a difference. Okay, any more alphabets? T, G, E, C. Okay, C is not good either. You're very tempted to want to use C here because you see crest, no crest, right? Which is quite a nice alphabet to complement, but not so good. Any more alphabets not to use? M depends on how you write M. Some of you may write M capital this way, then small caps this way, then it's fine. What are some alphabets that are really good to use? P. P. P also a possibility that you may misconstrue what is big and what is small. Unless, of course, you have lines demarcated. Because when you look at big P, small p, I know what differentiates caps and small caps is where the line is, right? Where the body of the P forms. But if we provide you a blank space, you may lose track of this yourself. What about Q? Q is okay. A, that's okay. Any more? B, thank you. B is okay. Any more? R, you get the trick? Then the next thing is, students always ask, what alphabet should I use? Is there a reason why this teacher used N? This teacher is okay. No reason. Okay, sometimes for your own sake, you want to be so imaginative and creative, you could follow what the first alphabet of the dominant trait is. For example, no crest is the dominant allele. You see it starts with alphabet N. You could use the alphabet N then. But if the first alphabet of the dominant trait is, for example, when we look at flower color, purple, white, then maybe I don't want to use P. Maybe I want to use A instead. So the truth is, any alphabet will do, but if you don't want to be so creative about it, you could follow the first alphabet of the dominant allele, or you could just, every question you do, just use big A, small a. Okay, that is fine. So that's the first part, you find your alleles. From then on, I'd like you to draw your attention to all the headers to the left. Okay, look at all the headers on the left. I'd like you to practice this. Write down all your headers first before you begin drawing a genetic diagram. Why? So that you don't miss out any line of this genetic diagram. Okay? So let's begin first with all the headers. And all these headers I hope make sense to you. The first header, parental generation. I'm going to short form all this, but you write it in full. So parental generation, make sure you underline it. This is a header. The next line you will go is parental phenotype. So we are starting to make use of all the vocabulary you've learned. Then parental genotype. Second, gametes. Then the next set of headers, F1 generation. This refers to the first filial generation or the first generation of offspring. And then you observe F1 genotype and F1 phenotype. Last but not least, expected phenotypic ratio. Again, write this in full later on. So, once all the titles are settled, this is where we can begin solving your puzzle. Often we have been solving our puzzles in our heads, but now we visually represent it so that you can justify to the assessor or whoever is looking at your justification that you have some logic behind your answer. So let's look at the question. Perhaps some of you already know how to fill this in. Let's go ahead and try. A pigeon which is heterozygous for the crest gene mates with another pigeon which is also heterozygous for the crest gene. Okay, let's begin. I have some blanks here that I'd like to fill up. Observe that there is a cross and a cross here. Yeah? Okay, these crosses have a meaning. They represent something that you have learned before in the previous chapter of reproduction. Fertilization. Okay? Fertilization. So, since this, maybe you see that later there will be some of our past friends. I'd like you to try now. We have the phenotype here and the genotype here that you have to fill in. I will start things off. This is where you can write down the genotype of the parents. What would the genotype of the parents be? Which of the three? Homozygous dominant, homozygous recessive, or heterozygous? Hey, our friends from Vietnam, they are already picking up. Heterozygous? Heterozygous, yeah? How would we write this out? So you will write big N, small n, mate with, fertilize, right? Or mating, big N, small n. Could you know based on your intuition? Oh, actually, we've already filled it in for you. Okay, big N, small n, big N, small n. Let's go backwards. What is the phenotype of the parents? Could you fill this in now? Because we did not provide that to you. Everyone write down the phenotype of the parents. Is it crest or no crest? And then we will just quickly run through. You will write down the phenotype. Okay. You're doing practice makes progress. Okay. Quite intuitive, heterozygous. So, how young? What were the phenotypes of the parents? Okay. How young says the phenotype of the parents is homozygous, dominant, and okay, homozygous, recessive. Any thoughts? Henry, what about you? What do you put? Okay, Henry, you're saying the phenotype is the characteristic. So what do you mean by that? Okay, for example, maybe no crest or crest. Okay. So what has Hao Yang proposed here? What is he suggesting here? If you say these are phenotypes, then what's Hao Yang presenting to us here? The characteristic of the phenotype. Then what is this? Oh, okay. What Hao Yang is saying describes the genotype. This is what phenotype is. So let's define. If you are still not very sure, maybe you want to write down some of these points. That's why I love genetic diagrams for beginners. It's a very good refresher of all the things we've learned. So what is a phenotype? Phenotype refers to the traits or the characteristics. Genotype refers to the combination of alleles. And if you notice the combination of alleles, there are a few ways you can represent it. On one hand, we can describe the genotype. So for example, if we look at this genotype here, how I would describe this genotype is that this is heterozygous. Now, this combination I see here is heterozygous. Okay? So therefore, maybe I follow up with Henry. Henry, then what is the phenotype of this genotype? No crest and no crest. We're building on what Ethan brought up earlier on, that no crest is the dominant one. So therefore, on the surface, it must be the allele that will show itself. Yeah? Everyone got no crest, no crest? Okay. Next comes all these lines. What do all these lines refer to? Okay, let's refer to the first set of lines. You observe that there are two lines branching out from each genotype. And then there are circles. I'd like you to highlight this particular process here, all the lines coming out, is referring to yet another chapter that came before, meiosis, in the topic of cell division. You see how elegant this model is? It strings together a lot of things from previous chapters. From cell division, meiosis, to fertilization in the topic of repro. It gives you a full picture of how one comes to be. Because in meiosis, remember, one cell will divide into the gametes, and the alleles will split up at the end of the day. And some of you are very quick to fill in the blanks already. Big N, small n, big N, small n. Each of the sex cells will get one copy of the allele. And that being said, what are all these circles? Back to the topic of repro, these are sex cells. Or more, or you know it as gametes, or your sperms and your eggs. This is in the topic of reproduction as well. And also a little bit of meiosis. The next part is the slightly tough part. You see all these lines over here, how might one draw all these lines? But I hope you appreciate what these lines represent. Hey, I made a mistake over here. These crosses over here shouldn't be fertilization. How can individuals fertilize each other? Okay, it doesn't make any sense. So let's remove this. What do you think the cross represents there? Sorry, Kara? Yes, it's great. But what do you need to do before reproduction can occur? Sorry? Sorry? Mating. Okay, mating. So, I got this wrong, huh? This is mating. Individuals can mate. The parents can mate. Yeah? Their sex cells cannot mate. When a sperm and egg comes together, they are not mating. Yeah? But, then when does fertilization occur? That's what all these lines are for. So, I'll teach you how to draw the lines because the lines look a little bit messy. First set of lines you can draw, I tend to go from sex cell to sex cell. So we go something like that. Huh? First set of lines you draw comes up from this particular sex cell here. You draw one line, then draw another one over here. Okay, that's the first set. What you can do now on your iPad is use highlighter. Use highlighter to color code the lines so you know where the source is. You don't confuse yourself. You see, I have a few colors here. Okay, let's go with these few colors. So blue. So this set of lines come from this first sex cell. Next set of lines you observe comes from this second sex cell. Let's use another color. We can now start from the extreme. Okay, we can start from the third one. Another combination here. Where does this one go? It goes all the way to the extreme left. We use the last color. Okay, I'm going to use blue again. Yeah, I think it doesn't crisscross too much. So the last one. the branching point of each one? Can you color code your lines right now so you know where the lines branch out from? And I think once you are done with that, the next most intuitive thing to do is, could you write down the genotypes of each possible combination of offspring? So I have a few blanks for you to fill in now. Can you write down the 4 genotypes, the 4 possible genotypes you can get from these 2 parents after fertilization? So all these lines here represent fertilization. When the sex cells come together as one again to form zygotes. Could everyone fill in these genotypes as well as the phenotypes? And then after that we will share with the class what your conclusions are.
I think for today, since it's crashing, it may be easier if you get a notepad. Yeah, then you just draw. Because then you can see.
Oh yeah, I think you got the drift already. Yeah. So what is the genotype of these 4 over here? Okay, big N, big N. N. Okay, next one. Big N, small n. Next one. Big N, small n. Both small n. How do you arrive at this? Follow the lines up. Because if you look at this line down here, it fertilizes this particular sex cell here. That's how you get a combination. This line here fertilizes this particular sex cell. That's how you got it. So now maybe you just continue on. What is the phenotype of these 4? Oh, okay, very good. Simple, no crest except for the last one. Okay, crest. Last but not least, I observed many of you who have already went to the last step because it's quite obvious. They ask for the expected phenotypic ratio. Indeed, they are asking for what's the ratio of the phenotypes. May I get you to just practice this? Will you write down no crest is to crest is 3:1. I know your practice is you have just written 3:1. But sometimes we don't know what the 3 is and what the 1 is. So put a small little header with ratio so that the teacher knows what the numbers are referring to. For all you know, maybe, this particular example is very obvious. But sometimes the examples we provide you may not be so obvious. What if it's a 1:1? And how? How will we know what you're referring to? Okay, so put down the header so we know what you're referring to. And so, this is your bread and butter for this chapter, being able to draw genetic diagrams. It is a very elegant model for beginners to refresh everything you've learned in the previous 3 topics, from reproduction to cell division to even fertilization, when sex cells come back together to form diploid organisms. Now, yes, Hiran? Okay, Hiran asked me to simplify the ratio. How will you simplify 3:1? Okay, fair enough. What if the ratio is 2:2? Yeah? Do we simplify the ratio? Yes, you can. Okay? In fact, you should simplify the ratio. It's 1:1. Okay? I think this is enough of us trialing. What you've been doing is to fill in the blanks. Now I'd like you to try it from scratch, from the stage of defining and choosing what alleles you want to use, to solve the next puzzle. But before we go on, have we solved this puzzle? what is the probability of getting an offspring with the non-crescent phenotype? Sorry? Sorry, yeah? 75%. Okay, they want for no crest, right? You know how you get 75%? 3 over 4. I understand the 3. How do you get the 4? Correct, right? There are 4 possibilities in total, right? So it is the 3/4, and that's how she got the ratio. So times 100%, that is the probability of an individual getting low crest. Is it possible for the first 3 eggs to be crested? I say again, is it possible? Let's say you have these 2 parents come together, they lay 4 eggs. They lay 4 eggs in total. 4 eggs. Is it possible for the 4 eggs to be all crested? Okay, who thinks it's possible for it all to be crested? Okay, Henry and Kyra, teacher, Alpha also. Who thinks that there's a possibility that it will appear as, 3 of the pigeons pop up, no crest. The last pigeon that pop out will have crest. Okay, who's in that camp? We listen to it. Kyra, you swap. Sorry? Isn't it what? Wouldn't all of them be? Also, my question is which one will be a possibility? So imagine these 2 pigeons, they lay 4 eggs. My question is, what will the babies turn out to be? 2 options are. The first one, that you will have 3 non-crested, you will have 1 crested. B, is it possible that all are crested? Is it possible, C, that all are non-crested? Which one will you choose? Sorry? Okay, our friend says, could this be more like a kind of like rolling the dice? You have this probability here. The probability is that 75% of the time you will get no crest. But is it possible that every time you roll the die, you hit 25%? You just nicely the 25% chance. Possible? Because every egg, you restart that probability. Every egg has that 25% chance or that 75% chance. Then what's the point of us doing this over here? What's the point of us knowing this 3:1? Well, this probability will show if the pigeons here have thousands of eggs. Okay? If there are thousands and thousands of eggs, this ratio will start to show. Probability-wise, you'll start to see that in 10,000 eggs, 25% of them likely will have no crest. Sorry, crested, 75% will have no crest. Okay? Yes, how yeah?
There's a chance that even if you have 1 million, they all strike the one.
Oh, could that be? Okay, that's a good question. So, Hoya asks, 4 eggs versus 1 million eggs. Okay? Is there a chance that 1 million eggs you are going to get 25, you're just going to hit the 25% 1 million times? What do you think? Possible? Hoya, if it's possible, I think you should go and try to, okay, no, I should not advocate gambling, but gambling is all about probabilities, right? If there's a small chance that you can win, and in 1 million times you always win, right? You make a very good gambler. Yeah? But it doesn't work out that way. The same reason how you look at genders. Actually, it's a 50% chance, right? Either male or female. But you look at how many of you are at home? All female siblings. Okay. All male. That's me, Who has male, female siblings at home? Okay? Will we observe this 50-50 if your parents had 1,000 kids? Okay. The higher the number of offspring you have, the more you start to observe this probability ratio. But the smaller the number of offsprings you have, unless you will see this probability. So thanks for bringing that question up, because it's all about numbers. The larger the sample size, the more you can see this probability. Smaller the sample size, you will not see it. And you see it in your daily life also, you look at your siblings, right? It's not 50-50. If that were the case, then it will be very easy for parents. First child come out, girl, then it's like, okay, I want a boy next, right? I'm going to have a child one more time. But then I know of some families want a boy, right? Then girl, 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 girl. Or they want a girl and then boy, 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 boy. Yeah? Okay, so it's all about the numbers. Let's practice now. Can you click on the second one? I would like you to try this puzzle. This puzzle only provides you this information. A heterozygote mates with a homozygote for the crest allele. The first question you want to ask yourself is this homozygote dominant or is it a homozygous recessive? How do you figure out? Okay, but the clue is they say homozygous for the crest allele. So it's the crest allele dominant or recessive. Okay, so you figure out. Then you're asking for the probability of getting an offspring with no crested phenotype. These are the steps, but you need not follow the steps if you find it comfortable to do it another way. But later on, we'll have a look at everybody's drawings. And then we'll try to pinpoint how we can improve on everyone's drawings or how we can learn from each of them. Okay, the time now is around 9:40. I'll give you around 10 minutes to try drawing your own genetic diagram from scratch. I want you to write all of this out, or you can type it out. I want you to draw these lines, draw all these symbols. You can choose your own symbol if you like to. If you want to reuse N. Okay, but we'll have a look at how your choices turn out to be. And then you solve the puzzle. Once done, can you submit it onto Google Classroom? And then we'll feature some in class. Yeah, today we go for Korean cafe. Thank you, Ella, for your class. Done already. Okay, so we'll feature some of your work in a while. Yes.
Oh, picture is facing one. But it's okay. Use a paper you can draw and then you take a photo. But I still want to check how you draw it. Like Rohit also, I can just check what you draw. Yeah. Okay, so fair enough. I agree with you. What you've done here is very elegant and fast way to solve the puzzle, right? Yeah. Yeah, I understand. So, I admit, when it comes to MCQs, please don't go and draw this entire diagram to solve a puzzle. Yeah? You can use just a square, because it is enough to solve a puzzle. What are these diagrams for, therefore? It is a way that we can see whether you understand the flow of the genetic story. Yeah? It is really a beginner's model. You don't find experts, researchers still using this model, but it's a very good tool for beginners to help you recall all the various steps. So, Yes, so we will explicitly say. We will say, we give you heterozygote, homozygote. Can you draw a genetic diagram to explain what is the probability? Correct. Yeah. However, nothing's stopping you from drawing this double check. What you order. I agree. I sometimes get lost in all the crisscross lines that I draw. Yeah, I get lost. Then I write the genotype wrongly. But I always draw this square in a corner somewhere in pencil just to double check. Yeah. But the experts don't use it.
Some of you are faster. If you are faster, go to practice makes progress. There are 3 challenging MCQs. Puzzles you can solve. Yeah.
So, let's see. This is a homozygote. How many alleles were there? No present match, right? So it's either 2 or 4. That's what it's saying.
Okay, I say again, you can go practice makes progress if you are done. I'll give you 5 more minutes, up to 55. 55 for you to touch up your genetic diagram. If you find it easier to draw on paper and take a photo, please do so. The intent here is to learn from each other. Practice makes progress. And if you're done with that, go ahead and play a little bit more genetics. Correct, yeah. The correction is always correct. Then sorry, the header is the phenotypic ratio. Pick up. Okay, about 2 more minutes. So, either practice makes progress, or you, and once done that, then you can go to genetics to play a little bit more. In a minute's time, we're going to have a look at some of your friends' work. Okay, let us review some of your friends' work. You be the teacher now, yeah? Imagine you are the teacher looking at the genetic diagram. Does this make sense to you? Okay, we're going to look at Zixuan's one. Okay, everyone have a look at Zixuan's genetic diagram. Okay, any feedback? What if it's sharp? Huh? Any of the steps missing? Okay, for Zuni, she says she will simplify the ratio. That is the practice you can do, simplify the ratio. So therefore, 1:1. Okay? So first thing, you could simplify the ratio. Anything else? Sorry? Anything else you spot that is not complete for the model? Okay, when she says, no, didn't define the alleles. I give benefit of the doubt, you were following the alleles from the previous definition, yeah? Okay, but you are expected to define the alleles. You need to tell me, let big N be the dominant allele for no crest, and so on and so forth. Okay? Anything else? Okay, underline the headers. Okay, so you've underlined the parental generation. It would be good to underline here as well. Okay, these are headers. Okay, the last one for myself, the mating crosses. So you look over here, we should have X's over here to represent mating. no mating, you cannot expect any fertilization to take place. Okay? But a lot of things, good things to keep note of. Look at Zixuan's drawing, extremely neat. You can tell line for line what corresponds to what. I look at F1 phenotype, it's all on the same line. Look at your drawing. Are you off the lines? Is it neat? Okay? So, I think for neatness, and actually I would say this is an 80%, 90% complete diagram. Okay, very good for first try. Zixuan has also answered that 50%. Yeah, that's right. Let's go to another example. Over here I see lots of colors. Who's this one? Okay, this one is Zixuan's one. Okay. Same thing. Any areas we can improve on? Neatness aside. Neatness aside, I expect you to use ruler, Ken. Don't use your free hand. When you do on paper. Any areas that we can work on? You all spot anything that we can work on? I like this one. They simplified the ratio. Yeah. Where are your headers? This 1 is 1. What is it referring to? Okay, so headers, important. Anything else? Kira, you want to say something? The what? It is, sorry? Stem 1. Oh, okay. So same as earlier, you need to define your alleles. Yeah? Again, I assume that you didn't define your alleles because it was previously defined. The question defined for you, that's fine. Okay? But you need to define your alleles. Other than that, if I scan across the board, this looks quite good. Okay? I'll just like draw your attention to the headers. One of these headers not properly defined. Can you tell which header? Zunyi, you identify it? This one? Okay. F1 gene. So, I'm not sure if this is you cut short the full spelling. But be careful. It is, as well as on the top, parental genetics. It is generation. Okay? We're referring to the generation. F1 generation. Parental generation. The I, okay, it's my fault. Copy me, go forward on the whiteboard. So it is F1 generation. Is it? Yeah? Parental generation. Perhaps if I shorten generation, it looks like gene. Shorten a little bit more, I don't know what you're feeling already. Okay? Let's look at some more. Okay, last one before we move on. Okay, maybe look at two more. So I see a lot more streaming in. So, okay, this one is very nicely colored. Let's see what Charmaine has prepared. Okay, any feedback on Charmaine's work? Another very organized and neat genetic diagram. Anything to improve on? Marks? Charmaine, I like that you have. Okay, hello, welcome. Okay, let's welcome three of our friends, also from Vietnam. Do you know each other? Oh, they know each other. Okay, let's go back to Charmaine's work. I don't know about you, I think this is quite a well done genetic diagram. We look at the headers. Let's use Charmaine's one as a recap then. Charmaine defined the alleles and she was a little bit more daring. She didn't go with what we worked with previously, which was N. She chose her own alphabet. Big A, small a. Let big A be the dominant allele for no crest. Very good. Trait. let small a be the recessive value for crest trait. Very well defined. Then she creates the two headers, and then she goes parental phenotype, followed by genotype, gametes. She circles the gametes. Very well done. All the meiosis lines, the fertilization lines, all done well. Everything is neatly arranged. They don't overlap with each other. She even simplifies the ratio at the bottom. Okay, this is a very good example. And I think across the board, I see that many of you are adopting the very good practice as well. Let's look at Shafiqa's one. Your file has been uploaded. You may proceed to try again. Okay, sorry Shafiqa, another day. Shafiqa's example is also good. Wait, hold on. Okay, I can zoom in here. Okay, Shafiqa also went on to define her alleles. Even though it were alleles we used before, big N, small n, she still went ahead to define it for us. You see the headers very well defined. All the headers filled in. She used very neat lines, circled her gametes, meiosis lines, fertilization lines, mating crosses, phenotypes well defined, genotypes well defined, genotypes, phenotypes, ratios. She also gives headers. This is a very good example as well. Okay? So I'd like you to adopt these practices. In truth, so you notice that the thing that changes between every question is what alleles you use, right? Because all these lines, these circles won't be the same. It is what you fill in the blanks with that matters. And therefore it goes back to your understanding of what's a homozygote, what's a heterozygote, what is dominant, what is recessive. This is the crux. This is merely a model. You can use the model as an effective tool, but at the end of the day, the concepts you need to know are what is going to ground you in all this. Okay? To end off this segment of the lesson, let's go to practice makes progress. We have 3 questions for you to try. I'd like to give you perhaps maybe 5 minutes, 5 to 10 minutes to scan through the questions. I'd like you to try the questions out. For those of you who are done a bit faster, you can go and play a bit of genetics, or, go to number 4, A4. You can start filling in some of your questions for me to answer before we close off this segment. Okay, so 2 things you can do. The 3 MCQs, as well as give me your questions for today. Okay, how are you? Oh, yes. Okay, let's begin. Give you just 5 minutes. I notice almost 3/4 of you have done the MCQs. So just 5 more minutes. You done MCQs? You can. My iPad working already. Yeah, I'm going to go through the MCQs in a minute's time. Hope you find some joy solving these puzzles. Yes. Go. Quickly go, then I'm going to go through the questions really. Okay, may I get everyone to go to the MCQ section? We're going to go through the MCQs now. I've scanned through your answers. I noticed we are split between two options. Okay, let's analyze this puzzle. Humans can inherit either brown eyes or blue eyes. Two brown-eyed parents have 3 blue-eyed children. Okay, 2 brown-eyed parents have 3 blue-eyed children. So here we have a set of parents, both brown eyes, and then they have children that are all blue eyes. All 3 of them. Wow, too many eyes to draw already. Okay, all 3 blue eyes. Sorry, I know you studied the chapter on nervous system, so you understand when they say blue eyes means it's the iris portion, not the eye blue color. Okay, that's very off. Okay, so 3 blue eyes, parents brown eye. It said which 2 statements are correct for this situation. This is challenging. It did not tell you which is dominant, which is recessive, right? But this is the fun of this chapter. Is you solve puzzles. Use your analysis skills to make sense of which one is dominant, which is recessive. Let's look at all the options now. Each parent has an allele for brown eye and an allele for blue eye. Is this possible? Let's look at the next one. The allele for blue eye is recessive. Wow, this is like, they are defining right now that the blue eye is recessive. Why can't it be dominant? Let's look at the first 2 statements first. Look at the last 2 later on. What makes you so confident that blue eyes is the recessive allele? Why can't it be brown eyes as the recessive allele? Okay, everyone have a thought. But some of you did get this question correct. What makes us so sure that blue eyes is the recessive allele? The way I like to solve this question is, you think the other way. What if blue eye were dominant? If blue eye were dominant, would the context of this question make any sense? So you're keeping a bit. Okay? So think both scenarios. If blue eye is recessive, does this question context make sense? If it's dominant, does it make sense? So is it a fair statement to say that blue eye is recessive? Okay. History up. Let's on it here, just in your own words up. What makes you say for certain that blue eyes have to be recessive allele?
Because the parents like their brown eye, and like if they had like if they only had like the brown eye, that means they can't even have blue eye children, so they have to be hetero, like
Okay. So Shreya is going one step further. She's saying that she thinks that the parents are likely heterozygotes, right? Okay, maybe I'll just use big A, small a. She says they have to be heterozygotes. Because she says that in the next generation, to have blue eyes means that they likely have to inherit one of the recessive alleles each to be able to get blue eye at the end of the day. Yeah? Okay, but my question also is, what makes us so certain that this allele here is blue eye, not brown? You're on the right track. But how might we justify to someone who doesn't believe you that blue eye cannot be the dominant allele? What if it were? Okay, Alpha says, because the eyes are brown. Why do you make that statement? Okay, Alpha says, if blue were the dominant allele, then the parent's eyes will be blue. But your term is brown, right? And yet, somehow in the next generation, pop out one blue. This is very classic of what we've encountered with the pigeons also, right? Or the purple and white flowers. Recessive alleles can kind of skip a generation sometimes because they are masked by the heterozygous genotype. Okay, so thank you, Alpha. It cannot be the dominant allele blue, because if it were the dominant allele, the parents would have blue eyes as well. Yeah? And I follow up with what Shreya said. Two heterozygotes, there's a chance you can even get blue eyes. But the parents confirm have to be brown. Okay? So statement 2, correct. Each parents have to have one brown eye allele, one blue eye allele. Yeah, has to be also. And if you notice, to get an offspring that is homozygous recessive, what you require is each parent to at least have one of the recessive allele. Because there's no way. If at least one parent is homozygous dominant, there's no way the offspring could have the blue eyes. Yeah? Now let's go to statement 3, because I noticed that is where we make the difference in choice. Right? We will split between A and B. So we look at statement 3. The probability that your next child will have blue eyes is 75%. Let's do the quick table. So I just want you to know, I know I taught you how to draw a genetic diagram, but when it comes to MCQ puzzles like this, don't go and draw the whole diagram out. You have no time. You know what's an easier way? Draw a square. Then you put in the alleles. And then you do a quick cross. Yeah? Okay? Don't go draw the whole diagram. You have no time for that. But we can see here, you will have 3 of them that will turn out brown eyes. 1 of them will turn out blue eyes. So what is the probability here? The probability to have blue eyes should be 25%. 1/4. Yeah? So I think perhaps the probability of your math went wrong. Last option. The probability that the next child has brown eyes is 50%. It is 75%, not 50%. Okay? So that's where we will split up between A and B. Let's go to the next question. Oh, this is a very interesting next question. Coco, many of us will split between a few options. We chose O. A lot of us chose B. And then a minority of us chose C, which is the correct answer. So we will kind of split between B and C. Let's look at the question closely. What could have tricked us here? Okay, they say this species of fish strength is controlled by 2 alleles, big B and small b. They say fish with the big B allele have very strong bones. Homozygous for the big B. Those who are heterozygous have thin bones, but weak. And those with the homozygous for small b do not grow in an embryo. Into an embryo, what does that mean? The baby doesn't even develop. It won't even come out of mommy. Won't even be attached to the mom with the via placenta. Sorry, it's a fish, so it won't even be attached to the yolk, won't come out in the egg. Okay, so 2 heterozygous fish mate. So big B, small b. Let's draw this square. And these are the possibilities. Fair enough? Now let's answer the question. What proportion of the offspring will be heterozygous? This is a very easy question, right? Okay, we observe there are 2 out of 4. The probability is half times 100%. It should be by right 50%. Yeah? Why is the answer instead 67%? Shafika? Okay, Shafika says if you look at this baby here, no count, even develop fully into term. So it won't even come out. The moment you get this, it is certain death. That means to say, in reality, your ratio goes like that. It is 2 out of 3. What is 2 out of 3? Yeah, my math, this is 66.6666 onwards, right? So it's 67%. So this is a trick question. Yeah? But the fact that you can choose 50% shows that you, at the very least, understand what heterozygous means. Let's look at the last question. Okay, last question. Okay, very similar. Brown eye with brown eye, 5 children. 3 with brown eyes, 2 with blue eyes. Which statement about this family is correct? 3 options, and some of us chose A, lots of us chose C. Let's have a look. Both blue-eyed children will have the same genotype and phenotype. Fair enough? Because if it's the recessive allele, there's only one combination that will work out to be blue. Let's go to the third option, because a lot of us chose this. The 3 brown-eyed children must be heterozygous for eye color. So let's substitute all of this with instead eye color, yeah? Okay, I'll try to use the same alphabet. So, 2 heterozygotes. This one you get brown eye, yeah? Brown eyes. Brown eyes. What are all the possibilities? These are the possibilities. Okay. They say the 3 brown-eyed children must be heterozygous for eye color. So, actually, what are the possible genotypes of an individual with brown eye? What are all the possible genotypes? It could be homozygous, dominant. It could also be heterozygous. So, to say that the 3 brown-eyed children must be heterozygous is a stretch, because it is possible, but not necessarily all the time. You find that you can also have individuals that are homozygous dominant that result in brown eye. So it's not a must. Okay? So these 3 MCQs are examples of puzzles that you can solve. Enjoy solving such puzzles? Enjoyable? Yeah? Okay, I'm happy you're nodding your head. Back then, when I was a student, I don't understand genetics. To the point, when I see these questions, I just roll the dice, like probability, A, B, C, D. Genetic diagrams even, I see the question, I don't even try, because I didn't appreciate. But if you enjoy solving these puzzles, actually, nothing to memorize, it's all about applying your concepts. Okay? Okay, last section today. Any questions for today? Okay, we have quite good questions. Let's look at Kyra's questions. Wow. Okay, let's have a look at Kyra's questions. Are lethal alleles, that means alleles that can kill you, naturally decided to be recessive? It's interesting, If so, why do they exist? Is it to discourage incest, to ensure genetic variety? Okay, I have to first say that in biology, there's nothing, biology does not discourage or encourage. A lot of it is grounded in science and chance, events. But let's go back to this question. Actually, Kyra, what made you come to this question to say that lethal alleles are recessive? What observation you make to come to this question? are diseases, is it? Okay. Okay, listen up. Kara observes when she goes online that a lot of recessive alleles end up in diseases. Right? So the question is, lethal alleles or the alleles that cause disease tend to be recessive? Not necessarily the case. There are alleles that cause diseases that can be dominant. You can imagine how disastrous that would be. At the same time, you observe there are some recessive alleles that don't cause anything bad at all. For example, you know the blood type, the allele for blood type O is recessive. But is it a bad thing? Not really. Unless you think mosquitoes biting you is a bad thing. Okay, but that one is another whole topic. So not necessarily. Are there any dominant lethal alleles? Oh, okay. Yes, there are some dominant lethal alleles. And sometimes you may find that they may not even develop to full term. Okay, especially if it's dominant. So if yes, are the what? Oh, and I just answered the question. Last one. Are all abnormal genes recessive alleles? Are there abnormal genes, defective genes that are dominant alleles? Kind of the same as the previous one. But I just want to say, this is a very interesting statement. Abnormal genes. What do you define as abnormal? Whether they are? Okay, whether they are defective. Then let's go back to the question of how do new alleles arise? For example, for humans, we have the allele for blood type O, we have the allele for blood type A, we have the allele for blood type B. How did so many versions of allele arise? Actually, this goes back to what we've learned last year about genetics, DNA. Do you remember the word mutations? Yeah? At the end of the day, what makes one allele different from another is down to mutations. Largely, they produce the same protein. But there are some changes in the sequences that result in slight changes in the protein. It's all down to mutations. In other words, we are all mutants. Yeah? The fact that we're all different is because we are all mutated. Yeah? We have alleles that are different from each other. So, is it always a bad thing? It's not. In fact, it could be a good thing. That's why we are all different. Yeah? Our mutations are what make us unique. We all have the we all have the gene that defines blood type, but we have different alleles for it. Therefore, different blood types. We all have the gene that defines eye color, but we have different versions of it. That's why we have different eye color, different skin color, different heights, so on and so forth. But our differences is what makes us unique as a human population. It would be very odd if we are all the same. Okay, that's the next question. Wow, actually, Kara is more. What else do you say? What's your boy? Kara. Oh, I understand. Kara, I thought you were submitting answers to one of the questions. You were submitting a question. But it's good, Is it possible to use genetic modification engineering to allow incest to take place by eliminating the possibility of two recessive alleles causing diseases? If so, what is the ethical stance of this kind of technology? Could such technology be applied to endangered species to ensure all the newborns? Okay. Kara, if I summarize your question, are you asking could we apply genetic engineering to reduce the negative impacts of incestuous mating? Kara, I also want to bring up something. When you talk about ethicality, I'm feeling you are thinking a little bit, are you about humans? Okay, about humans. But I'd like to bring up another point for your consideration. Is it ethical for you to inbreed animals or plants? You're not shaking your head. But if you notice, if you go to the supermarket, voila, all your apples almost look the same. The corn that you buy, you want to buy this corn because it's consistently sweet. Yeah? Why is it so consistent? One of the reasons is because there's a lot of inbreeding. We inbreed so that we can get the same traits over and over again. The pigs we eat, the milk we drink, the consistent standard, part of it is attributed to inbreeding. Is that ethical? Okay, so that's a question to ponder. Human-wise, is it ethical? I think it also boils down to culture. It boils down also to what people believe in. Ethicality aside, is there a way to address genetically issues that may arise from incestuous mating, animal, plant, or human? I'm not sure if we've reached that stage yet, technology-wise. Okay, I'm not sure if we've reached that stage yet. But in our next chapter, we are learning about genetic engineering. As we learn the steps, you tell me, do you think at certain stages if it's possible for us to swap out alleles? Okay, is it possible? Let's say we can. Should we? Why would you? That's another question. And at least, I'm very clear at least on the plant and animal side. Why farmers do it is because we want to maintain traits that we define as good. Very rarely we want to inbreed if the traits are not good. And so if you want to question the ethicality of it would be, then if we do it for humans, who defines which alleles are good, which alleles are bad? Yeah? For all we know, I like people who are taller. Some people like people who are smaller size, fun size. Huh? Some people like hair that is black, some that is brown. Who are bad? How do we define? So perhaps that is the question you want to think about too. Who plays that role then to define what is good and what is bad? But one thing's for certain, when it comes to nature, no one defines what's good and bad. You know what defines what alleles will persist in the population? Nature. If you have a bad allele and you don't survive in nature, you will die. And you will not go on to the next generation. So in nature, environmental factors define it. Okay, is that 31? Today is your math teacher or CCA? Okay. So I will close out today. Next session, we'll address some more of your questions. Before we go into blood types, so today we learned about joint genetic diagrams. Next session is all about blood types. Then you can also solve genetic puzzles and perhaps even find out what your parents' blood types, genotypes are. Okay? So that's all for today. Thank you class and thank you for joining us today. Okay, thank you.